Hello friends, today we are going to solve AS Biology Paper 3 Advanced Practical Skills 970033 October November 2023. So let us move on to this first question. So yeast cell contain enzymes that hydrolyze sucrose into reducing sugar as shown in figure 1.1. So they have given the equation in which they clearly mention sucrose which is getting converted to reducing sugar with the help of the enzyme that is present in yeast cell. So what is the question they are asking us to do here is we need to investigate the activity of the enzyme in yeast cell. So yeast cell will be immobilized in sodium alginate beads. So the sodium alginate beads is nothing but immobilizing the enzyme. So immobilizing enzymes we are going to do. So what are the setup they have given to us is we have been given with yeast cell suspension, sodium alginate solution, calcium chloride, sucrose solution and Benedict solution. Um, Main thing when we are doing this experiment, if any of the harmful irritant that comes in contact with your skin, please wash off with cold water immediately. So in this experiment, we are asked to investigate the activity of yeast enzyme by using different numbers of beads of immobilized yeast cells in the sucrose solution. Yes. So to carry out this, we need to follow the steps, whatever they have given. So as a first step, they asked us to take 10 centimeter cube of A into a beaker. So A is sodium alginate. From the given stock, we need to take 10 centimeter cube in a separate beaker. Then Y is the yeast suspension which is given in a beaker. That from the yeast suspension stock we need to stir it, and then we need to take 10 centimeter cube of Y from the beaker, and we need to mix that with the beaker containing A, that is sodium alginate. So we need to mix both yeast suspension and the sodium alginate together. Then we need to take 20 centimeter cube of C in another beaker. C is calcium chloride. So we need to use 10 centimeter cube syringe to collect 10 centimeter cube of mixture of A and Y. So the mixture of both sodium alginate and yeast suspension should be taken together in a 10 centimeter cube syringe. Then we need to hold the syringe over the beaker containing 20 centimeter cube of calcium chloride, which is shown in this figure 1.2. How it is shown in the image, we need to hold it in such a way so that when we are holding it with one hand, we need to gently press the plunger so that drop by drop along yeast suspension and the sodium alginate will be added to the calcium chloride solution, which forms the bead. So this should be done repeatedly so that we need to at least make 31 beads. So, and finally the immobilized beads are left in beaker for 5 minutes to make them to retain their proper shape. uh, After 5 minutes we need to take out the beads, and then we need to transfer it to the petri dish. So we need to test this activity of yeast enzymes by using different number of beads. So they have also given how many number of beads we should use. So they have mentioned it as 1, 2, 4, 8 and 16 in a sucrose solution. So next one, so we need to carry out the steps 10 to 20. So step 10, first we need to label the beakers 1, 2, 4, 8 and 16, and label 5 test tubes 1, 2, 4, 8 and 16. So as they have given in the image, we need to label the beaker and then add that much amount of beads into that beaker. Then we need to put 10 centimeter cube of sucrose solution S into each beaker, and then we need to start the timing and then wait for 5 minutes. So while we are waiting for this time, we can set up the water bath to do the Benedict's test. As this is going to reduce the sucrose into reducing sugar, we need to check with Benedict solution. So Benedict solution requires heating, so the water bath can be set up during this time. So in step 19, we will be using the water bath to check with the Benedict solution. Now the question asked here is, state the temperature you will use for water bath. So the temperature what we can use for water bath is the 80 degrees Celsius or more. So anything above 80 degrees Celsius is good for testing Benedict solution. Then heat the water bath to the temperature that have been stated. Then you are going to at the end of 5 minutes stir the contents of each beaker. Then we are going to use syringe to transfer 2 centimeter cube of solution from beaker 1 into the test tube labeled 1, and we are going to repeat this step for other beakers which contain 2, 4, 8 and 16 beads in the same way. So taking 2 centimeter cube of the solution from the beaker and then adding it to the respectively labeled test tubes. Then we need to put 2 centimeter cube of Benedict solution B into each of the test tubes that have been labeled. Then we need to put one test tube in water bath at a time, and we need to start the timing, and we need to check how long it takes for the appearance of the first color change. If the color change is not appearing after 2 minutes, so we need to stop the timer and then record it as more than 120, and we need to record all our results in the given table. So appropriate table should be drawn for this result. So to draw this table, we should know two things, that we have to mention the independent value and the dependent value. So here the independent value is the number of beats, and time depend on the number of beats that are present in the solution. So keeping that in mind, so if you are going to draw the table, number of beats will be 1, 2, 4, 8 and 16, and time will be 110, 94, 73, 54, 44. All these timings are just the example what I have written this for our solving purpose. So when we are doing the experiment, actually the time might vary, and so the time here is inversely proportional. As the number of beats increases, time taken will be decreasing. And always we should know that whenever we are writing something related to time in seconds, the time should be in whole number. Now, moving on to the next question, state the independent variable in this investigation. So the independent variable used in this investigation is the number of beats. So based on the number of beats, the time varied. So here the independent variable is the number of beats. Then state one main source of error in this investigation. So one main source, size of beats are not same, that is the major error. Then not all beats are left in sucrose for the same time period. So the different time periods were there. So these two are these two are the example what is the source of error. From this, any one can be written. Now moving on to question 5. A student set up a beaker as a control experiment. The result of the control experiment showed that the sucrose was hydrolyzed by an enzyme. Suggest what substance the student put in the beaker for the control experiment. So control experiment should be boiled yeast. So if yeast is there, the hydrolysis is taking place and sucrose will be converted to reducing sugar. But if you are going to add water, we cannot confirmly tell that yeast is the main reason for converting this sucrose to reducing sugar. So we need to add boiled yeast, which tells that clearly that dead yeast cell does not convert any sucrose to reducing sugar. So boiled yeast is the answer. So next question, the procedure described in step 1 to step 20 investigated the effect of changing the number of yeast beats on the rate of hydrolysis of sucrose. Now we are asked to describe how you would modify the procedure to investigate the effect of changing the concentration of sucrose solution on the rate of hydrolysis of sucrose. So what will happen, or how to modify this procedure based on the modifying the concentration of the sucrose solution for identifying the rate of hydrolysis of sucrose. So to investigate the effect of changing concentration of sucrose, we need to at least use 5 concentration of sucrose solution. So this 5 different concentration of sucrose solutions can be prepared by using serial dilution method or simple dilution method. And one thing to be maintained constant is the bead number. So here we are not allowed to change the bead number, as we are checking the hydrolysis of sucrose based on the concentration of the sucrose solution, not based on the number of beads. So next one, pectinase are enzymes that are used to increase the volume of fruit juice extract from apples. Pectinase can be immobilized and placed inside a container with apple pulp as shown in figure 1.4. The fruit juice is then collected. Then the scientists carried out an investigation to determine the effect of temperature on the activity of immobilized pectinase by measuring the volume of fruit juice extracted. All the variables were kept constant and the results were shown in the table 1.2. So this is the table 1.2, which shows the results for immobilized pectinase at different temperature and the volume of fruit juice collected. So we are given temperature from 30 to 80, and respectively the volume of fruit juice collected was also given. So for this we are asked to plot a graph. So how to plot a graph? So for plotting a graph, first we should know what should be written on the x-axis and y-axis. X-axis should always have the independent variable and y-axis will have the dependent variable. In this case, temperature is the independent variable and volume of fruit juice is the dependent variable. So we need to um, scale the graph. So I have scaled it as 0, 30, 60 and 90 for x-axis for with temperature. Then for y-axis I have taken 0, 20, 40, 60, 80 and 100. So with respective values we are supposed to plot the graph. So all the graphs should be plotted, and I have not drawn any connecting lines with the plotted points. So you can use your scale to connect the points point to point, or you can just draw freehand line, curved line. So this gives the graph. Then use your graph in figure 1.5 to estimate the volume of fruit juice extracted at 55 degrees Celsius. So exactly at 55 degrees Celsius, what will be the volume of fruit juice extracted, they are asking. So from my graph, I come to a conclusion that it is at 90 centimeter cube. So here is the reference graph for you. So this tells that at 55 degrees Celsius the amount of fruit juice collected is 90 centimeter cube. So the blue line mark states that. Then describe the change in volume of fruit juice shown by your graph in figure 1.5. So from 30 to 60 degrees Celsius the volume of fruit juice increases. After 60 degrees Celsius the volume of fruit juice decreases. Then the scientists repeated the investigation, replacing the immobilized pectinase with the same amount of pectinase but not immobilized in beets. This time the scientist has used free pectinase. So again they have given the temperature and the volume of fruit juice for free pectinase. So what are the question asked? Suggest an explanation for why more fruit juice was obtained when using the free pectinase between the temperatures 30 degrees Celsius and 50 degrees Celsius than when using an immobilized pectinase. So when more apple, like when the enzyme is free, more apple pulp can come in contact with free enzyme than with the enzyme immobilized in beets. Then suggest and explain for why more fruit juice was obtained when using immobilized pectinase between the temperature of 60 degrees Celsius and 70 degrees Celsius than when using the free pectinase. So free enzymes denatured more quickly than immobilized enzyme. So this immobilized enzymes are capable of withstanding higher temperatures. So at 60 and 70 degrees Celsius the activity of pectinase was still seen, and more amount of fruit juice were obtained when immobilized pectinase were used at 60 and 70 degrees Celsius. Moving on to second question. So we have been given with slide K1, which is through a which is a plant leaf, transfer section of plant leaf, and we are asked to draw a plant diagram, and we are asked to label and identify epidermis. So the diagram whatever I have drawn here is the outline or the general diagram of any leaf structure, but according to the slide whatever you are seeing, you are supposed to look into that slide and then draw the actual structure of the plant diagram. A plant diagram actually have only the lines and no individual cells should be drawn. So epidermis, the top layer or at the bottom layer, both are upper and lower epidermis. Any epidermis can be marked since they have not mentioned any specific epidermis whether upper or lower. So you can mark the epidermis in any place. So I have taken the upper epidermis and then I have marked this. Then second question, observe the cells in the epidermis of the leaf on K1. Select a line of 4 adjacent epidermal cells. Each cell must touch at least one of the other epidermal cell. Then make a large drawing of this line of 4 cells. Use one rule, label line and label to identify the cell wall. So we are supposed to draw a single line of 4 adjacent epidermal cell which need to be touch one another, like each cell should touch at least one other cell, and we should mark a label line to identify cell wall. So to do this we can just take any 4 cells from the epidermis with high power lens. High power lens in the sense it is around 40x magnification, 40x magnification objective lens. We can take 4 adjacent epidermal cell, and then we are supposed to draw this, which looks like this. So 4 epidermal cells, and then it should have all the double membrane structure to clearly show the difference between the cell wall and the cell membrane. And one important thing, no broken line should be there. So this I have drawn with my tab, and there are few broken lines, but when we are drawing in paper, exam paper, it should be clearly drawn in a single line without any break in between. And cell wall should be marked very clearly, and the label line should touch only the cell wall. It should not go inside or touch the cell membrane and then labeled as cell wall, which will be marked as wrong. Next question. So figure 2.1 is a photomicrograph of stained transverse section of a leaf from a different type of plant. So this is a different type of plant which is not there in there on the slide. So we need to identify 3 observable differences between the leaf on K1 and leaf shown in figure 2.1. So record these 3 observable differences in an appropriate table. So again here they have mentioned we need to draw a table. So we need to draw a table for 3 different features. So here we need to mention what is the feature we are going to compare in both slide K1 as well as in figure 2.1. So here I have taken 3 features, like most of the time the 3 features what comes are the number of vascular bundle present. So we should just check how many vascular bundles are present. So in this particular experiment, since I have seen the slide, so I know that it has only one vascular bundle, so I have written it as 1. And in this figure we can able to see there are many vascular bundles, so I have written many. And the size of vascular bundle in slide K1 it is small, and in figure 2.1 it is large. And size of xylem also, it is small in the slide, the specimen in the slide, and large in the image given here, the figure 2.1 image given here. And epidermis, so the epidermis, the epidermal layer is also thin on the specimen, and here it is thick. So these are not only pointers what we should see for the features. Other than the number of vascular bundle, size of vascular bundle, or size of xylem, epidermis, we should also check for trichomes, whether trichomes are present, and arrangement of vascular bundle, how the vascular bundles are arranged, whether it is arranged in a pattern or it is randomly arranged. And airspace, presence of airspace or absence of airspace, that can also be written as a feature. Moving on to next question. So figure 2.2 is an enlarged version of the transverse section of the leaf shown in figure 2.1. So it's an enlarged image. We are asked to measure line P and line Q to find the length of vascular bundle and the length of vascular bundle P and length of vascular bundle Q as shown in figure 2.1. So here we simply need to measure the length and then write it with appropriate units. So you can either write it in centimeter or millimeter like this. So yeah, so we are just need to measure it with the scale and then write the correct value with the proper units. Moving on to next question. Calculate the percentage difference in the length between vascular bundle P and vascular bundle Q. So we are asked to identify the percentage difference in the length. So, and we are also we should also give our answer in 2 significant figures. To calculate this percentage difference, so percentage difference is Q minus P by Q into 100, so which is 49 minus 29 divided by 49 into 100, which is 40.8%, which is equal to 41 percentage. So with this we are completing this paper. I hope you people will find this helpful. Please do like, subscribe, and share, and give your queries and thoughts in the comment section. So let me know what to improve and how to improve other stuffs. Whatever you want, you can just tell me. Or if you want to know more about any of the other concepts, like conceptual learning, if you have any doubt, you can also mention that. I'm ready to take those concepts in the upcoming videos. Thank you.